Then we come to variations of the PCR, and we talked about some of them yesterday, and we talk about a little more today. And so colony PCR, I've already told you, colony PCR basically implies that instead of isolating the DNA and then amplifying, you directly take the colony, denature it, and directly dip it into the colony and do a PCR. So, and then of course you have nested PCR, multiplex PCR. So let me come to these individually here. So this is your colony PCR. The screening of bacterial E. coli or yeast clones for correct ligation or plasmid products can be checked using colony PCR. They pick a bacterial colony with an autoclave toothpick, swirl into 25 microliters of tea autoclave double distilled water in a microfuse tube. Heat it to boiling water, so you heat it to 100 degrees centigrade for 2 minutes, and then the spin soul sample for 2 minutes at high centrifuge so that the debris settle down, and what you have is the DNA. Transfer 20 microliters of supernatant into a new centrifuge, and from this you can take up 1 to 2 microliters of supernatant and add it as a template in a 25 microliter PCR tube for a standard PCR reaction. So you're trying to basically do a boiling lysis of the bacterial cells, and after the boiling lysis you are taking that particular material and dipping your toothpick into it, and then of course you are adding a bit of that liquid into your PCR mix. Then of course you could have what is known as hot start PCR. I already told you hot start PCR is to increase the specificity of primer binding and getting a specific amplifications. So if you are getting non-specific amplification, you can follow hot start. And in hot start, what you do is you first denature for 10 minutes, and after that you add your Taq polymerase. And nowadays, of course, you have automatic Taq polymerases that get activated only after the initial denaturation is done. So therefore, this gets easy again. So the technique may be performed manually by heating the reaction components to the melting temperature, 95 degrees centigrade, before adding the Taq polymerase. So first you have the reaction mix ready, the PCR mix, and then you heat it to 95 degrees centigrade for 10 minutes, and after that you add your Taq polymerase. Specialized enzyme systems have been developed that inhibit the polymerase's activity at ambient temperature. Ambient means room temperature or slightly higher, either by binding to of an antibody or by presence of covalently bound inhibitors that only dissociate after a high temperature activation step. So when it is 94 for 10 minutes, this inhibitor is going to deactivate, and now your enzyme gets active. Then of course, DNA polymerase that you use as most commonly what you use is Taq polymerase, but you could also use PFU or Vent and other polymerases. Then the thermophilic DNA polymerases show a very small polymerase activity in room temperature. So these are basically at room temperature, they do not really get activated. Asymmetric PCR, yes, well, in case you want to specifically amplify only one strand of the two strands, then you can basically what you can do is you can use a lot more concentration of the strand whose amplification you want. So of the two primers, either the forward or the reverse primer is in small concentration, and the other primer is in excess. So that allows for asymmetric amplification of one strand as compared to the other strand. And sometimes you may require that especially for sequencing or hybridization, you may need this. So asymmetrical PCR is used to preferentially amplify one strand of the original DNA more than the other. It finds use in some types of sequencing and hybridization probing where having only one of the two complementary strands is ideal. PCR is carried out as usual, but with the great excess of one primer as compared to the other. So this is your asymmetric PCR. Then you have nested PCR. Nested PCR is when you want a very specific product and you want it in a very highly purified form. So what you do is you do a two-step PCR. First, you design an outer primer, and from the larger template, you amplify with the outer primer, getting a specific product now. So this is your product that you get from the outer primers. And now you do an internal primer, which is slightly inside of the outer primer. So you take up this amplicon now, and now you do a second round of PCR using internal primers. So what you get is a very, very distinct and something that is non-contaminated or non-spurious DNA of the region that you wanted to specifically amplify. There is nothing that comes from elsewhere, because from the genomic DNA, you have taken up a slightly shorter target now, and from the shorter target, you are now doing the amplification. Two pairs instead of one pair of PCR primers are used to amplify the fragment. First pair amplify the fragment similar to standard PCR. Second pair of primers, nested primers, as they lie are nested within the first fragment. So this is the first fragment. Now second pair of primers are within the first fragment, and that is giving you a nested PCR, right? Second PCR product, which is shorter than the first one. Advantage, very low probability of non-specific amplification. So whenever you want very specific amplification and you want to be absolutely sure that the product that you have is what you wanted, you do a nested PCR. So this is 2 rounds of PCR. You do a first round of filtering. From the larger genomic DNA, you are amplifying a very specific region. And from this specific region, you are re-amplifying the region of your interest. So this is 2 layers of 2 rounds of PCR, and that gives you a very specific thing. So then, of course, what you can do is amplified fragment length polymorphism. So amplified fragment length polymorphism, you know now. So basically what you do is you take the genomic DNA and then first fragment it. So in this case, AFLP and RFLP, what comes first is done second. So AFLP, amplified fragment length polymorphism, first you fragment the DNA. So here is your DNA. You fragment it with MSP1 and EcoR1. So here you are. You basically do, this is your genomic DNA. You digest with MSE1 and EcoR1. And then, of course, after you've done your digestion, you add adapters to them, right? So here is your adapters. Basically, these are your, if you remember now, adapters are partially double-stranded, right? So they have one end that is sticky end and the other end that is basically carrying sticky ends. But in this case here, you add the adapters here. So once the adapters are added from the sticky end, because they have complementary sticky ends, so they can add here. And then, of course, what you do is you do an amplification using the sequence of the adapter and moving inside into this this region where you want to amplify. So basically what you've done is your amplifier, you have first digested your DNA. When you've digested your DNA, you create a library of fragments. From this library of fragments, you want to amplify specifically a subset of fragments. Because if you amplify using the adapter sequence, then what would happen is everything else will get amplified and then again you'll get a smear. What you want is some distinct bands. So for that, what you do is you design primers that are complementary to your adapter, but also move inside into your fragment. Let's say here in this case you have, so this is your adapted sequence here until ATT, and then what you've given is 3 extra nucleotides, G, T, and A, right? And when you give these 3 extra nucleotides, only the fragments that correspond, that have a sequence of CAT here will be amplified. So from a large library of fragments, you are only amplifying fragments that have at the 5' end a CAT sequence. Likewise, for the reverse primer, you have added a GCT sequence. So only those that have a GCT sequence are going to be amplified. So specifically from a library of fragments, you are amplifying the fragments that at the 5' end on one strand have GTA and on the other 5' end on the complementary strand have a CGA. So basically you are now amplifying a subset of fragments. So double digest, ligate adapters have complementary sticky ends at 3' end and prime complementary sequence at the 5' end, right? And then amplify with the specific primers. To amplify only a subset of fragments formed, the primers may have 1, 2, or 3 extra nucleotides shown in black. So this is your 1, 2, and 3 extra nucleotides. When you do 3, you amplify a very small subset. If you make 4, you're still amplifying a much smaller subset. 5, even more smaller subset. So the more you extend it here, the more you now are specifying that I want something that begins with GTA and then also has a G and then also has a C. So now only the fragments that have a 5' end GTA, GC would get amplified. We take this example a little further. So here you are. So this is basically, let's say the arrows indicate your restriction sites. So you do a complete digestion, double digestion, you get these fragments. To these fragments, you do the adapter ligation, and after the adapter ligation, you do a first pre-amplification with just 1 extra nucleotide in the primer apart from the adapter. So now, again, you are amplifying just a subset. And from this, you do a second round of selective amplification where you now have 2 or 3 extra nucleotides. So from this subset, again, you're picking up subset of amplification. And that then allows you to amplify specific fragments that correspond to a specific size. So when you do a sub-fractionation of the library, you will be able to see distinct bands. That is the advantage, because you don't want a smear. So what you want to do is to amplify the library of fragments that you've got, but only a specific and select fragments and not every fragment, so that you get a clear banding pattern and not a smear. So when you do that, this is what you get here. So if you see here, there are 4 panels here. Each panel shows 3 EcoR1 MSC fingerprints using 3 different primer combinations. Human DNA fingerprints are displayed in panel 4. So this is your panel 4, and if you see here, this is 10, 11, and 12. So 10th one, you have EcoR1, and then of course you have CAC MSC. So basically you have this EcoR1 sequence, and then it has this 3 nucleotide sequence, which is CAC, and this one here is basically the MSC1 adapter. So you have MSC1 adapter sequence and CGA. And then of course in the second one, you still have the same EcoR1 plus CAC MSC. Here in this case, you change the 3 nucleotides to CAA. And then in third case, you have CAG and CGA. So if you see, accordingly, the pattern of bands that you get changes out, right? So, and this is another. So the fourth one is human genome panel. Then of course, if you look at the other ones displayed in panel 1 is Arabidopsis. So Arabidopsis 1, 2, 3, these are the primer combinations that we use for amplification. And based on the primer combination you have used, different fragments will show up differently here. Then of course, you have tomato in the second panel, and you have maize in the third panel, and you have human in the fourth panel. So this can be used now for developing a fingerprint of an individual, right? So that is again important, because now you're moving your primer 1 to 3 positions, and if the third position of the primer is polymorphic, I may get a band because in my DNA that particular nucleotide is complementary to the primer. In somebody else, where this position is polymorphic, and the sequence is not polymorphic, you will not get a band in that individual. So this can be used to generate a DNA fingerprint. Remember that, this is important again. That DNA fingerprinting can be generated using amplified fragment length polymorphisms, and as the name indicates, polymorphism means you'll have variation in length of the fragment that you've got. So if you see here, depending on the primer you choose and depending on the individual you choose, you may have different fragments because each carry a specific variation in their DNA. So however, still tedious, involving digestion, ligation, and PCR. So AFLP, while it is useful, it is really tedious because it involves first the digestion of the DNA, then ligation of the DNA with the adapter, and then of course the PCR. And this is the dominant marker type because here you cannot distinguish between homozygotes and heterozygotes. And I'll show you an example of a co-dominant marker type as we move along. So molecular weight range of fingerprint is from 40 to 45 to 500 nucleotides is what you get here. This is one thing. Then of course, advantages of AFLP, multiple polymorphic restriction sites can be screened in one go. So if you have multiple polymorphic sites which you want to screen, you will be able to use AFLP to do that. The bands are scored for absence and presence. Unique individual profiles with it. So this is the major advantage, that individual profiles which are unique to each individual can be developed this way and help in unambiguous identification of crime suspects or settle paternity disputes. So this is basically the advantage. Disadvantage, it is a 3-step process. You start with digestion, then you have to ligate, then you have to do a PCR. Bands from different regions may co-migrate to the same distance because of the similar size confounding the analysis. So sometimes what may happen is that you may see a band between 2 samples at the same position, but they will not actually be representing the same sequence. So because of the size, they have come up the same position, but they may be coming from 2 different regions of the genome, and therefore you may easily get confused that these 2 are identical while they are not. So therefore you move on with other things. And then you have what is known as randomly amplified polymorphic DNA. This is again a dominant marker system, which means that you're not specifically targeting a specific region in the genome. You're doing a genome-wide analysis. And here in this case, you want your primers to go and join and anneal to the DNA and random positions. For that, what you do is you use short primers. So these are single primer. One, you use a single primer which serves both as forward primer and reverse primer. This is specifically the uniqueness of this primer is that it is a short primer. It is not more than 10 nucleotides long, and because it is just 10 nucleotides, the chances of it having a complementary region on the DNA are much higher at a lot many positions. So here I show you one random primer that I'm using, and because it is complementary to this region, it goes and binds here. Another primer on the opposite strand comes and binds here, and now they are within the PCR amplifiable distance of each other. So this would give a amplicon of 300 bases. Likewise, another primer has come and bound to another region of the genome here, and reverse primer is basically the same primer in the opposite strand has also come and bound because it found this strand it found a sequence which is complementary to it here. And now this can give you 100 base pair banding pattern. This can give you 500 bases and so on and so forth. So used when no prior information about the genome is available. So the question is, now that we have so much of sequence information about genomes, why should we use it? So we don't really use it now. This was a technology that was used roughly 15, 20 years ago, and that was when we did not have enough sequence information for anything. So therefore, because you do not have any sequence information to begin with, you cannot design primers complementary to a sequence you do not know. So therefore, what we used was random primers, and from here, then once you get the bands, you can sequence them, get some sequence information, and now design specific primers for this. So this is a randomly amplified polymorphic DNA. It generates very beautiful patterns, and I did a lot of rapids when I was doing my PhD. So this is one example of a randomly amplified polymorphic DNA. So I had mouse from where I used to pull out the DNA. So this is from the same mouse: brain, heart, kidney, liver, spleen, and testis DNA was pulled out. And then this is another set of mouse where again I take brain, heart, kidney, liver, spleen, and testis. And these are your PCR markers. So ladder, this is the 1 kb ladder, this is a 100 base pair ladder. And then you see when you do your amplification, you get these patterns of bands, and because it is coming from the same individual and different organs, if you see very clearly, you have exactly the similar pattern of DNA here, right? PCR banding here. So these are the multiple regions where my primer has gone and amplified specific regions here. Each of them has a different size, so you very clearly see 1, 2, 3, 4, 5, 6, 7, 8, around 8 bands are here. Likewise, in this mouse, this is a different mouse, and here again, if you see, you get a different banding pattern. So, and the problem with rapids was that because you are allowing for random amplification of DNA, and the primer is going and annealing to the DNA pattern in a random fashion, because of its small size, it is able to find a complementary sequence more frequently in the DNA, and it goes and binds there and starts giving amplification. So therefore, because of the randomness of the system, rapids are not reproducible. And one of the primary requirements of an experiment that you want to take forward and you want to say that I can base my conclusions on this is that the experiment should be reproducible every time it is done. So therefore, rapids are not a very good choice. But then when there was no information available, you still use rapids. Then you could have an inverse PCR. So basically what you do is here is your restriction digest. You self-ligate the plasmid and then you design primers that are oppositely oriented to each other. So instead of one primer here and one primer here, which is the normal course, what we do is we design primers that are oriented in opposite directions. So this goes and amplifies this whole thing, and this goes and amplifies this whole thing. So finally you get a sequence that you can sequence here. So inverse PCR uses standard PCR primers oriented in the reverse direction of the usual orientation. So usually what you have is one primer is facing towards the right, the other primer is facing towards the, so the one primer is facing towards right, the left primer or the forward primer, and the reverse primer is facing towards the left, and both are moving towards each other. So in this case, what you're doing is you basically take a template, you anneal your primers in the center, and what you do is you circularize this DNA so that you can still get the amplification. The template for reverse primer is a restriction fragment that has been self-ligated. Inverse PCR functions to amplify sequences flanking a known sequence. Applications, amplification and identification of sequences flanking transposable elements, and the identification of genomic inserts can be done using this. Then multiplex PCR. Multiplex PCR is simple. In one PCR, you are using multiple primers. Only condition is their melting temperature should be within the same range of each other. So they would go to your template DNA and amplify specific regions, which would be seen as multiple bands in your PCR. So here is your individual amplification of the DNA using this. So this is a MEK 310 base pairs. This is LUKS FPV. This is 433 bases. There's 16 sRNA here, and what you've done is these are done in 3 different reactions of PCR. Here you have put all these 6 primers. So there are 3 products, which means there will be 1 forward and 1 reverse for each. So you put all the 6 together in one single tube, and with the same template you have amplified, because the primer conditions, extension, and everything are exactly the same for all the 3 set of primers here. So multiplex PCR enables simultaneous amplification of multiple targets of interest in one reaction by using more than one pair of primers. So all primers must be specific and must have similar TM. So the condition for multiplex PCR is, so you can save time, you can get your amplifications done in a single reaction, provided that all the primer sets have nearly the same TM and all the primers are highly specific and amplify only their own region and nothing else. So that is something that you need to keep in mind. Then of course you can have long range PCR. So normal PCR, I have told you, not more than 2 kb can be amplified with normal tag polymerase. But there are mix and there are proprietary enzymes that can amplify you much longer length of DNA. And when I was in my PhD, I did what is known as triple master from Thermo Scientific, and that used to be the one that I used to use because I used to amplify around 22 kb of sequence. So extended or longer than standard PCR, making over 5 kilobases. This is basically frequently over 10 kb. Long PCR is useful only if it is accurate. Thus, a special mixture of proficient polymerases along with accurate polymerases such as PFU are often mixed together. Application to clone large gene fragments not possible with conventional PCR. So if you want to amplify large gene fragments, you have to first do this thing. Then coming to reverse transcriptase PCR, we have already talked of this when we talked of making the cDNA library. So here you are. So this is your mRNA. What you do is you have an oligo-DT primer because almost all mRNAs would have a poly A tail. So this is a universal primer that can be used to amplify all the mRNA molecules present in a RNA mix. So you do you add an oligo-DT and then you add reverse transcriptase and dNTPs. Reverse transcriptase is going to use RNA as a template and form a DNA molecule that would basically have the DNA nucleotide. So this is your DNA RNA hybrid that you have first, and then of course mRNA can be degraded with alkali, and then you can synthesize the second strand. So finally you have your reverse transcriptase PCR. So we have done this also when you're doing cDNA and synthesis in the previous lectures in libraries. So based on the process of reverse transcription, which reverse transcribes RNA into DNA and was initially isolated from retroviruses. So this is reverse transcriptase, reverse transcribes RNA to DNA. The first step of RT-PCR, first strand reaction, is the synthesis of cDNA using oligo-DT primer. So what you do is you use oligo-DT 37 degrees centigrade for 1 hour, and you get reverse transcriptase, and you'll be able to synthesize the first strand. The second strand reaction, digestion of cDNA RNA hybrid using RNase H, and the standard RNA PCR with standard PCR with DNA oligo primers. So basically, this is what you do. Allows the detection of even rare or low copy mRNA sequences by amplifying its complementary DNA. So low copy mRNAs are not easily identified, and for that you need to do a reverse transcriptase PCR, convert it into cDNA and amplify it, and then of course you'll be able to identify these low copy PCRs. Then allele-specific PCR or ARMS PCR or amplification refractory mutation system. So this is a PCR that is used to basically identify your SNPs, and what you do is basically you design primers until the SNP position. So the 3' end of the primer is basically complementary to the SNP that you're looking for. So let's say if you have here, let's say if you have an individual here, so this is the position where you have the SNP, and the normal individuals have thymine here. So the other end you'll have an adenine, and then of course, so this is your 3'. This is your 5' here. So what you do is you design a primer, and then you have another individual in whose DNA you have, instead of thymine, you have an adenine at this position. So this position is polymorphic, right? So what you're doing is you're designing a primer here, and you're giving, so let me see if I can remove this a bit. I had an eraser here, everything gets cleared. This is bad. Anyways, so you have this, and you have a thymine here, and then you have another individual in which this position is polymorphic, and he has a, let's say, cytosine here. So when you give a primer that has adenine at its last base, so this is your 3' end of the primer. So therefore, now if you see, this primer will be able to amplify using a reverse primer which is on the opposite strand. you'll be able to amplify this piece of DNA. But for this individual who has a cytosine here, this primer that has an adenine will not bind here. And because it doesn't bind here, you do not get amplification for this individual. So this can be used to discriminate between individuals to distinguish individuals who have SNPs and the others who do not have it. So basically it is used to identify single nucleotide polymorphisms. You understand the difference between polymorphism and mutation class? Difference between polymorphism and mutation? All right, so Vibha says yes. So what's the difference? All right, so the difference between polymorphism and mutation is that when a mutation is at a frequency higher than 1% in the population, then it is called polymorphism, right? So it is basically frequency dependent. And of course, if you see now, you can do the PCR. So it requires prior knowledge of the DNA sequence, including differences between alleles. So you're looking for two alleles here. One allele has a thymine, the other allele has a cytosine because of the SNP. So you can basically design primer which is complementary to one of the alleles that will get amplified, the other will not get amplified. Use primers whose 3' end encompass the SNP. So the 3' end would end up at the SNP, and the primer would be complementary to one of the alleles present at this SNP position and not to the other. PCR amplification under stringent conditions is much less efficient in the presence of a mismatch between template and primer. Successful amplification with an SNP specific primer signals presence of specific SNP in the sequence. So this finishes our types of PCR, and now we stop here, and tomorrow we do expression vectors, and we finish the course tomorrow.